Okay, I think we'll make a start. What I'd like to do in about 30 minutes, which I've never done this before, is actually discuss, or I plan to discuss, the basic science, animal models and clinical rationale for the use of probiotics, the utilization of probiotics in health and disease. And this is going to take the form of an overview in terms of the interaction of commensal bacteria with the gastrointestinal tract. We'll also briefly look at the interaction of pathogenic bacteria and just briefly overview the hygiene hypothesis. I'd like to mention the probiotics, the hope, and also discuss objectively the hype of probiotics, because I don't think that they're going to cure every disease and I don't think it's going to prolong life indefinitely. I'll touch on probiotics and aging, and then if there's time permits, I'll just briefly discuss stress and its role in gastrointestinal tract. Okay, now there's a definition for probiotics, and a Dr. Fuller about 20 years ago defined probiotics as living organisms in food and dietary supplements which upon ingestion improve the health of the host beyond their inherent basic nutrition. And what we think probiotics are is that they're bacteria and other organisms that when ingested may actually treat or prevent disease. Now, if you look at the gastrointestinal tract, the stomach in a normal person is usually sterile, unless a patient's on on an acid blocker. The low pH in the stomach of about 1 is a very nice barrier to ingested microbes. But as you move into the small intestine, the proximal part of the small bowel, you can see that there's a rapid increase in the number of bacterial cells, which are around 100,000 bacteria per milliliter of fluid. And as you move into the proximal bowel, this increases to about 100 million bacteria per milliliter of fluid. And then as you move into the colon, there's another sharp increase, which is around 100 billion bacteria per milliliter of fluid. Now, it stands to scientific rationale and reasoning that if you have got this kind of a load of bacteria in the gastrointestinal tract, that there must be some sort of interaction with the host. From birth, there's an assault on mucosal surfaces. In utero, mammals are sterile, but on coming into the world, we recognise that there are 4 stages of colonization. And as I said, intrauterine, the baby's germ-free. Phase 1 is an acquisition of maternal vaginal colonic flora, all the way down to phase 4, where the individual acquires a complete adult colonization, usually by about 24 months of life. Now, there is a physiological change as the baby moves from this scenario to this scenario, and hopefully a little bit less aggressive, that at birth, what we've got is facultative anaerobes with a sharp increase, things like E. coli and streptococci, and then they decrease some once the baby moves from this scenario to solid foods with obligate anaerobes such as Bacteroides and bifidobacteria. Now, the gut physiology is a complex ecosystem. This is a very complicated microbiome. There are complex interrelationships going on there, and there are over 500 different kinds of bacterial species that we've recognised. It is associated with numerous physiological functions of the microflora. It's associated with barrier function and restoration thereof. The immune system stimulation, this is very, very important, because if the baby is not colonised with the right bacterial flora, this fails to fully develop. Maintenance of mucosa nutrition, production of nutrients, bioavailability of things, short-chain fatty acids such as butyrate, which we've actually shown at UQ to influence significantly liver fatty acid metabolism, and the formation of other things such as vitamins, the stimulation of bowel motility. But in relationship to the stimulation of the immune system, this gut physiology may promote immunological tolerance, which is a lack of immune system reactivity. Now, we really don't have fingerprints profiles of what really is a good stool, gut profile, gut bacterial profile versus a bad one. What the scientists tell us is that there are harmful versus beneficial bacteria. And if you look at things like lactobacilli and bifidobacteria, you know, they're reported to stimulate the immune system, inhibit harmful bacteria. And what we also know is that, you know, things like Clostridia and Pseudomonas and, you know, Staphylococci produce toxins that, you know, actually are harmful to the host. So therefore, this argues significantly that there is some sort of interaction with the gut, and the microbial physiology thereof can actually be, we can understand in terms of an immune reactivity why there are pro-inflammatory and anti-inflammatory cytokines. As for example, in the terminal ileum, you know, the development of Crohn's disease, or things such as constipation in the colon, or the development of ulcerative colitis in the colon, being an unbalance of beneficial and harmful bacteria. Now, how might commensal bacteria promote tolerance to bacterial and dietary antigens? Well, they do this firstly by inhibiting the growth of immunostimulatory bacteria, inhibiting production of pro-inflammatory cytokines by epithelial cells, promoting the generation of regular T lymphocytes, and actually strengthening the intestinal mucosal barrier by actually stimulating mucus and antibody production. And over the next 3 or 4 slides, this is basically what I want to show you. And we start with this very elegant experiment conducted at Washington University by Laura Hooper and her colleagues, where they looked at germ-free mice. And what they did is they inoculated them with this particular microbe, Theta iota omicron, which is a bifidobacteria. It actually adheres to the enterocyte, and what it then does, the scientists, what they then do is they go and have a look at the gene stimulation, and they tease out the effects of this bacteria in the development of the enterocytes, the transport of nutrients, and the properties that are associated with immunoprotection, such as increasing the levels of IgA, increasing the IgA receptors, and other proteins that actually inhibit the decay of the mucosa, of the gut mucosa. Along similar lines, you know, another experiment where in germ-free mice, again, looking at the effect of commensal bacteria interacting with these TLRs. These are toll-like receptors. They are pattern recognition receptors. They recognize dietary antigens or bacterial antigens and then respond accordingly. And if you look at the microhistology, you can recognize the Peyer's patches, you know, the germinal center, the dendritic cell. The dendritic cell here in the gut is acting like a traffic cop. It extends into the mucus layer in the luminal part of the mucus layer, and is actually signalling, taking up antigen, dietary or bacterial, and coordinating the immune reactivity response. And as you see in this particular experiment, you've got the TLR9 receptor, a commensal CPG, and that should be up here, so I apologise. It's a cytidine phosphoguanosine oligonucleotide, forming a vesicle that then produces T regulatory cells, promoting the tolerance and down-regulating inflammation. It's basically common sense. If the body's going to respond to an invader by producing pro-inflammatory cytokines, you need a way to bring it down, to maintain regulation. And commensal bacteria, probiotics, is what they help do. Now, there are a number of TLR receptors that are recognised. I've just put a couple here, TLR2 and TLR6, which are associated with microbial lipoproteins. This is specific for gram-positive, and this is specific for gram-negative bacteria, just to show you that there's a few of them around. This is a diagrammatic representation of basically what is really going on. You know, the lactobacilli working in the terminal ileum, and the bifidobacteria in the colon, and there's the dendritic cell, you know, organising the network between a Th1 and a Th2 response to maintain this regulated pro-inflammatory, anti-inflammatory balance. And the balance looks, I'll show you what it looks like in a minute. This other particular experiment is very, very interesting because it's looking at commensal bacteria in cell cultures, you know, their effect of incubating these kind of strains with cell culture enterocytes. And you can see that by themselves, without being inoculated, they produce some mucus. But when you actually incubate them with different strains of Lactobacillus strains, you can see that there's a significant increase in production of mucus. This argues very, very strongly that cells don't turn on and off by themselves. there's a signalling process going on, a very, very strong signalling process to maintain a regulated state in terms of a response.